In 2012, I became increasingly discouraged by the pace at which science was moving to advance Lyme disease research. And I was aware of the Institute of Systems Biology here in Seattle, and I was always really fascinated by the type of science that they do.
I started the Institute for Systems Biology in 2000. The idea was this was an institute focused on a brand new way of looking at biology. Rather than studying genes as we did in the past in a reductionistic manner, one gene and one protein at a time, systems approaches are global and holistic. They try and understand the whole system and how it operates.
I knew in 15 minutes when I was there, I said, this is the place. I can just feel it. Everything was already in place. There was a proteomics department. There was a genomics department. They had been a fully functioning, really high-tech science facility that had been, generating top-notch science for a couple decades.
And they're not afraid of the complexity of it. Because
Right.
I think when you first told me about it, I was like, this is the same, nothing's going to work. There's no, what are we going to do? And then we went in and he was excited about the complexity. He's like, this is my kind of puzzle. Let's do this,
About 4 years ago, Jeff and Liesl Wilkie came to me and were curious about just what systems biology was. And we explained it, had a wonderful conversation. And at the end, Jeff said, Liesl has chronic Lyme disease. We're looking for brand new approaches to attacking Lyme disease. Would your institute be interested? We actually set up a meeting where we invited Lyme experts from all over the country to come in Jeff and Liesl's home and talked about the possibilities. And at the end of that, we agreed that it would be an exciting idea to take a systems approach to Lyme disease. And I persuaded 5 or 6 of my major colleagues here at ISB to take, again, this global, holistic approach.
So when I came, I thought, there's a real opportunity to take some of the tools that we've been developing and take some of the concepts of scientific wellness, and as we move this Lyme study into a phase 2, really begin to capture this disease at a personal level, at a really comprehensive level, in a way that lets us understand why some patients go on to chronic disease and some patients respond rapidly to antibiotic therapies. And out of that work, maybe to begin extracting ways to treat those patients. Understanding how different organs are affected and what have you. And so as we've been planning this next phase of our Lyme program, we've been doing it with, I think, a really heightened ambition on what we think is possible now.
So what we've done is defined in interesting new ways the genome and its nature, and we've completely defined the proteome, all the proteins the organism makes. There are about 1,000 of them. And we have an atlas that's available to the public of scientists, and they can use that information to study Lyme disease in any way they wish. From this first 3-year study with the Wilkies, and then later on, the Cone Foundation joined the Wilkies, and it became the Wilkie-Cone Consortium attacking Lyme disease. From those studies, probably the most important thing we've discovered is 6 protein biomarkers in human blood that let you unerringly distinguish normal individuals from those that have been infected from Lyme disease. And we've discovered these, and we've actually confirmed them and validated them.
Problem is that the diagnostics are not that good. If you don't come down with the regular symptoms, how do you actually tell you have Lyme disease? Because the current diagnostics utilize the immune response, the human immune response, to detect that you actually got that, you have to actually wait for at least a month before you amount enough of an immune response to detect that. Problem is then you're living with that disease for quite some time. Other people will invariably be not diagnosed with that, so they escape this therapy, and so they end up going for this long term. And then there's another class of people which they class as chronic Lyme disease. These are people that have these lifelong symptoms, yet they do not have, none of the diagnostics actually tell that they still have Borrelia at this point. So one of the things that we're trying to do, at least to fix the first component, is to create a diagnostic at the very earliest stage and not look at the human immune response, but look specifically for the bacteria, and looking at it in a way that other people haven't looked at it before. What people have looked at previously is to look at proteins in their whole structure, but what we're looking at is remnants. So when the bacteria is inside your body, it doesn't live forever. It has to die too. It replicates, but the parents die at some stage, and that's what we're looking for. We're looking for those remnants of the bacteria as it's being then expelled from the body. And so we're looking for small peptide components that are non-immunogenic but are still getting expelled from the body. It's very, very small, very tiny amounts, but we need to look with those technologies to get to them.
Let me say also another thing that we did that I think is really striking is we recruited an immunologist here at the Institute who was able to study the human immune response to the infection. And she's come up with quite an interesting immune signature, again, that distinguishes the acute Lyme patients from their normal counterparts and everything.
The hypothesis that we're working with is there some sort of immune dysregulation in these individuals that leads them to manifest these symptoms? And one of the reasons why that hypothesis is so attractive is that if you look at other autoimmune diseases, things like SLE, MS, for the longest time they went misdiagnosed. And they have incredibly heterogeneous manifestations across different individuals. An issue that we also see with Lyme patients, again, that's comparing things on the surface.
Individual humans are really, really complicated organisms. And when they are infected by an organism like the Lyme bacteria, Borrelia, their responses are really complicated. And to understand both of those dimensions of complexity and the response, we need to do what is called dense phenotyping. That is, we need to literally make on each individual billions of measurements and to follow these measurements over time.
So dense data clouds is basically the notion that with modern technologies, we can make thousands of measurements about the body. And this is, many of these have only become possible in the last few years. And so as you think about it, if you generate a dense amount of data, you can think about the way that you do discovery and the way that you deliver medicine very differently. So if you go into the doctor today, in most practices you get a very small amount of data. It's generally a handful of measurements, and basically and they make some inferences off, and you go from there. The idea behind dense data is that we can make tons of measurements. So we can get billions of data points from the genome. We can get thousands of data points from proteins and metabolites out of the blood. You can measure things like the microbiome, these microbes that live in your gut. And the idea of taking all that information together is you can start to have a much more comprehensive and much more individualized view of what's happening in a particular person's body and how that's relevant to a particular person's health condition.
And in fact, I compare these dense data clouds with the Hubble telescope. It let us see the universe with a resolution never had before, and we discovered all sorts of things about the creation of Earth. our dense data clouds are going to give us a view of biology and of disease we've never ever had before.
So, over the past really 5 years, you couldn't have done this 3 years ago, we've developed methods that allow us to look at the repertoire of immune cells at the sort of detailed molecular analysis you can get, and to begin tracking out the infection history of a given patient. And you can do that now for a couple of common viruses. But with this particular patient cohort that we'll be studying this year, we ought to be able to do it for Lyme. And that means that if you've got a patient that comes in maybe half decade after they were initially infected, you ought to be able to look in their blood and read out that acquired immune response, that immune memory of the pathogen being present.
For us, we want to complete what we're doing in terms of finding markers for Borrelia in human clinical samples within the next 2 to 3 years. That's our expectation, that we will have specific markers that we can say we have validated in a number of people from samples from these people that have Borrelia or Lyme disease, that we can say, yes, we can detect this marker.
Lee and you and I were sitting at our table, at our kitchen table,
Okay, yes.
talking about this. And Lee kept saying that the biggest challenge in figuring this out was finding the right control and treatment groups, and then being able to afford to follow them before a Lyme season, so before people became infected, and then all the way through the season where some of those folks were going to be infected and treated, and then see what happened afterward. And trying to sort of be able to afford to do this without it being such a scattershot that you couldn't come up with any real results was very difficult. And Lee and I were just talking about it, and we said, why don't we just find a town that has a very high likelihood of people being infected because it's in the Northeast,
Right.
and see if we can convince some medical professionals in the town to help us just collect blood from as many people as possible before the season starts, and then monitor them through the season so that we can see the changes in their blood as some of them become infected. And we had the amazing good fortune to talk to Keith Frankel and
Right,
and Rocco about
right around the same time.
Yeah.
Yeah.
About their own struggles and progress in trying to help all the people that they knew in the Nantucket area who were affected by this.
Right.
And it's just come together remarkably fast. It's great to see ISB partnering with the professionals who are trying so hard to reduce the suffering in New England. And we're really optimistic about what's going to happen this summer.
Yeah.
So that's a study that we're working on with Dr. Rocco Monto at Nantucket. And there's a couple of other folks that are involved at Keith Frankel there. The Wilkies have been very helpful in this. We've talked about it with the Cohen Foundation. So let me sort of lay out what Nantucket's like. I was just there last week trying to plan the study. So I think everybody knows Nantucket is an island off of the coast of the U.S. It's about as far east as you can possibly go. It turns out that it's an island that is probably 80% rural, and then there's houses that are in communities. The point being, and we actually took a tour of the island so that we could understand how this is the laboratory of Lyme. It is actually staggering how many people on this island have Lyme disease. But you've got basically the houses and the natural environment of the ticks just coexist. There's not like a clean segregation. Even in the town of, I guess the Nantucket Town Center, I guess that's what it's called, it's a pretty small town, and there's a huge population of deer on this very tiny island that would just waltz through the town all the time. So we heard about Nantucket and about Dr. Monto and Keith Frankel from the the Wilkies, about probably 6 months ago. And we touched base with Dr. Monto and Keith Frankel, and we talked about what might be possible. And initially we were thinking we'll take maybe 50 patients in the first year and do a deep, very deep dive on those patients and then accelerate our program in the outlying years. And then we further want to look at about 100 patients that have chronic Lyme, and of course that's not an issue there. There's lots and lots of patients that have chronic Lyme, plus controls. And so we went last week and talked with some of the community leaders, the board leadership in the hospital, some of the physicians, and we had an incredibly warm reception. People are very, very excited about this project. And so right now we're pushing forward the IRB as fast as we can. It's got to go through the Mass General Hospital system, but so that we're ready to go come Lyme season this year. It's my belief that a year from now we'll be sitting here and we'll have a much deeper insight into, for an individual patient, what is their trajectory once they get bitten by a tick and take on the Borrelia pathogen. What is that trajectory over time as they either, maybe they have an internal resistance. There are certainly patients like that. Maybe they're going to respond well to the standard antibiotic therapy. We'll have a golden opportunity to try to standardize those treatments across the different physicians there, at least to some extent. Maybe they'll move on to chronic Lyme and we'll be able to see those transitions. And I think that will give us, for one thing, I think it's just intrinsically a scientifically rich question to say, why do some patients react? Why do you get the spectrum of responses? And you should absolutely be able to predict that.
We think there's a lot of reasons that we should, as a society, really care about what's happening with Lyme disease. Its incidence has been on the rise, and it's really important that we figure out ways to marshal the resources, both in people and funds and so forth, so that we can really make a difference in Lyme disease. But there are a lot of approaches that I think have emerged over the last few years that have real hope for this disease. And we're really hopeful about the future and that we can turn and stem the tide of Lyme that's happening across the country.